Okay, well, thank you for coming to this impromptu talk. When I agreed to come here in week 2, I didn't know that all the talks would be in week 1. Then I was informed about it when it was too late to change anything. So last week I was in London on another engagement. So let me tell you a story that has already been some years in the making, and the reason I'm telling you this story is that I still believe this is state of the art, and in case you want to use technology like this, the statistical physics framework for predicting sort of nucleosome positioning on mesoscopic scale, then we could talk afterwards, and we could, maybe exchange ideas and even software. So I also should say that I'm at Rutgers University in the US, and my appointment is in the Department of Physics and Astronomy, which was not the case when I came in, actually. It's a long story, but now I'm fully in the physics department, and this, of course, affects the types of things I do and the types of data and, just ideas I have access to. Okay, so I like showing this, even though it really means nothing, but my student made it a long time ago now. This is just to say that if you have, you take each chromosome and each cell, and they're all from a few millimeters to a few centimeters, and then you stitch them all together per cell. And then do it for each cell, of which there's 36 trillion in a human body. Then in an adult human body, then you get kind of an astronomical number, right? So you can have enough to go to the sun and back about 300 times, which is kind of amazing because all of this fits into your body, which is not of astronomical dimensions. 10%. Yeah, if you multiply this distance maybe times 2 or something like this, times 300. So that's kind of crazy, but this is, of course, no one is extending DNA molecules like this and stitching them together. Of course, physics and astronomy. Well, most people in bio, even outside bio in the physics department, they would let you be, but they wouldn't really know what you're doing. So this is an old figure from Gary Felsenfeld's Nature. And these parts are showing, multi-scale chromatin structure in Pekin. So you guys must have seen it hundreds of times. This is questionable, right? The more down you go, the more questionable it is. And now there are all kinds of new ideas floating around with loop extrusion and whatnot. But we're going to be safely here where it is okay, even though I will argue. So here is just DNA, and here's the bits on the string, right? This is nucleosomes. Even though I will argue that even this is kind of questionable, not always true, like a well-defined linker length. Okay, so we'll focus on these two levels, and you can see these bits on the screen here. These are nucleosomes. This is, I believe, electron microscopy, an old picture. This is just to illustrate that the idea that the system we study, I'll show it in the next slide. Actually, this is one of the nice crystal 1KX5 nice crystal structures for the nucleosome core particle. We all know the dimensions and so on, that it's 1 3/4 turns, and it packages 147 base pairs of DNA. Here's the picture I was alluding to. So this is kind of the mesoscopic scale, right? So what you want is maybe a few nucleosomes over the gene, and then a few more upstream, and so that the total number is maybe 7 or 10. So we're not studying thousands of nucleosomes, or not always. But what you want is also transcription factors and all kinds of chromatin remodelers, and ideally you want both dynamics and thermodynamics, right? So you want to study this in detail. And one big assumption that you make is that the three-dimensional contacts are only implicitly modeled. You're not modeling a fiber. I believe, to my surprise, actually, there was Wilma Olsen here last week. And she must have talked about Monte Carlo simulations of a short piece of fiber with actual three-dimensional conformations and stuff like that. This is what she does. She's in the same university, that's why I'm talking about her. We're not doing this. Instead, we're using a set of techniques from statistical physics that were originally developed by Perkus and others on one-dimensional liquids to develop, sort of nucleosome positioning in one-dimensional systems. So, okay, this is just to say that we're interested in dynamics as well. Transcription factor might come in and activate the gene, right? You might have to displace the nucleosome or unwrap part of it. So one thing that I wanted to emphasize is that nucleosomes are dynamic. So I'll give you some beta numbers, but this is a particle which kind of unwraps the ends, so that if you're a transcription factor and you need to come in here, you just have to wait, and then you could bind and sort of rectify this. And this could even happen one at a time, then another one could come in a little bit further in and so on. So this is some data from John Widham's lab that shows that indeed unwrapping happens. This is the nucleosome covered dissection enzyme sites, and you can basically, the height here is the accessibility. So they're accessible on the ends and less accessible, but even somewhat accessible towards the center. So here is a slide where I've assembled a few order of magnitude estimates, which come from different people, some of which were here last week, and are based on models and the number of experiments, just to know sort of what the energetics of nucleosome formation is. So you have actually not just a uniform path for DNA to bend, but you have patches. So you should think about it as, you unstick a patch and then it opens, one turn opens, and then you have another one, and so on. So people have measured, like of the type I showed just now, on the previous slide, the accessibility to restriction enzymes, and this estimates into like a couple KT. KT is 0.6 kcal per mole per patch, and this argues for like 21, 28 KT per nucleosome. So if you did, of course, you neglect, I just took this number, which comes from accessibility assays, and I multiplied it by 14. Okay, so long before that, the nucleosome would probably disassemble, lose the outer parts and so on. But if you sort of neglect that. So there is single molecule nucleosome unzipping. There's much more now, but I left this number, which is from Ohio State, Ralph Bunschuch. So this kind of corresponds, so 601 sequence is a good folder, right? So it's like binds tightly. It was designed by John Widham specifically to create stable nucleosomes. And this shows analysis of this experiment, which is a single molecule unzipping experiment, is 24. So it's kind of roughly the same. So, but this is like a special one. I forget whether it was, I guess this could have been 601 or similar as well. But Widham also did competition of his 601 to genomic DNA from chicken. And he argued in that paper that compared to this number, which is special because this nucleosome was bred or evolved for binding affinity. So the typical nucleosome should be weaker by about 6 kT. So it's like 23 minus 6. Okay, so what do we take out of it? So it's not easy to unfold nucleosome, right? It's still much bigger than kT. You need about 1 ATP to do this. But by the same token, it's not a stable particle with 147, right? Because to unwrap like the two outer patches is just a few kTs. So even thermal fluctuations can do this. So the same paper actually estimated also the range. The range means like the weakest binder to the strongest binder. And it puts some sort of sigma on this. So it's basically, but this is just guesswork, especially for this one. But very roughly, it's like 18 KT for chicken nucleosome plus minus 4 KT. Okay, maybe less than 4 KT, 22 is already pretty stable. Maybe there are a few like this. This is the total energy. Yeah, so this is the total energy, and that's a good point, because if you take a worm-like chain and bend, that's about 60 kT. So, but that neglects, there's like this is free energies. This is just a bending energy, so there's loss of entropy and other effects. So this is just total, and it's mostly experiment-driven. One number here might be more model-driven. Here they've, you do this force extension curves, and then you do the area, and then you get the work, for example. So they're not read off directly, but they're kind of reliable. Okay, so, so all this, even if I believe this number is only approximately, all of this tells me that, I have to have nucleosomes as mobile particles because they will unwrap. They might translocate. This is probably slow if it's unassisted. They will not unfold easily. And ideally, you have to have several, that's for biology, right? You have to have transcription factors and nucleosomes, right? To just model your mesoscopic system better. Yeah? Yeah? Yes, of 601, yeah. Mm-hmm. Yeah, I don't know. It's a nucleic acid research paper from about 15 years ago. Michel Wan did the experiment and Ralph analyzed the data. Yeah. Okay, so I thought that was I thought they were doing what you said first, just pulling and unzipping. They may have done both. So what I'm getting out of here is an order of magnitude estimates, which argue for the mobility, or limits of mobility of the nucleosome and how far it's willing to unwrap under thermal fluctuations. So this argues for some sort of statistical mechanics framework. This is a thermodynamics model I'll show today. Dynamics is still under construction. It's quite a step forward to do kinetics of this thing. But basically, we wanted to have the following ingredients, and they're all available in the model as it currently stands. We want to have unwrapping rather than a fixed size. We can have a fixed size particle as a special case. Then we would like to have multiple species competing for DNA access. We would like to have some sequence dependence for nucleosomes or, in fact, transcription factors, if we wanted to. And then we wanted what's called, in physics, potential barriers and wells. This is like tightly bound particles, for example, that you don't want to model explicitly, but want to basically model as a boundary condition, as a wall. Many early simulations were done against the wall, so we wanted to have that functionality as well, where the nucleosomes self-organize. And then we would like to have effective two-body interactions. People usually don't put that in, but this is to reflect Linker discretization patterns, for example. Meaning there is a 3D structure imposes some constraints on linker lengths, and you can put that in implicitly in a 1D model. I will show to you some more pictures. This is the subtlest effect, and this is the hardest to model, but it is there. Okay, so here is just the argument for sequence dependence, which is to say that at least some people believe that some dinucleotides could go where the minor groove faces the molecule and then 5 base pairs later, it should be like this. We're not married specifically to any of these models, but the sequence dependence is there. If, for example, one uses machine learning to find out new features, those could be easily put in. So this is my idea of barriers and wells. For example, RNA polymerase or whatever, it binds tightly, and nucleosomes have to organize left and right of this sort of mountain that's tightly bound in a certain way, right? So that they find the minimum of free energy. So this is my argument for this linker discretization. These are not necessarily, they're highly simplified models. This is 1-start, 2-start business. But what I want to say is that if you have some sort of 3D chromatin fiber, even if it changes with time, right? But you have sort of transient, maybe 3D structure, then it will, or it may impose some constraints on the length of linkers, and we can actually put that in. So this is all I argue for. These are some old simulations that show some fibers. Basically, it doesn't matter. I'm not arguing for any of these fibers to be true. I'm just saying that there may be transient or even persistent effects that that translate into effective two-body interactions by restricting linker length. Okay, so there are 2 ways to solve this problem, and we did both of these ways. So one is a direct problem, which is you give me the energies, you tell me which components are important, and I give you the occupancies. So this is running the algorithm forward, right? You can not only have occupancies, but correlation functions, given that something is at position I, what is at position I plus delta? But you do have to give me energies. So it goes from, or free energies, from energies to particles, distributions of particles. And the way we envision it is kind of like this. So this is DNA, you bind, you have steric exclusion, right? Which is what makes it, if it was just an ideal gas, that wouldn't make, it would be a simple solution, but you have steric exclusion, and you have 1-body binding and 2-body interactions between nearest neighbors. Okay, so if you have this guy may talk to this guy, but if there was another one, then that would be 0. So there's a nearest neighbor interaction. And they're necessarily short range, so even though we haven't pushed the length, but they can be extended maybe by 20 bases, like 20 bases away. So we define the 1-body binding energy of a particle that sits on DNA, and then the 2-body interaction between nearest neighbors. And forward solution, this is just a technical detail. It just tells me that the particles can be between A min and A max. So each particle can have a range of lengths. Okay, this is to model nucleosome unwrapping. Of course, for transcription factors, maybe you don't want that, but for nucleosomes, you do. So formally, it's done by putting energies to infinity if they're outside the range, and then there is a sequence-dependent energy within the range. So the same is for the interaction is translationally invariant, so it depends on k minus j, where j is particle 1 and k particle 2. So this is the 2-body effect, and what you want to compute is what's in physics called a grand canonical partition function. So you don't have the control of the number of particles on DNA. You basically have constituents in solution, and then they bind however they want to, and this is controlled by the chemical potential. So of course, this is a huge, so the whole trick is what I'm not gonna show. This is so we've found efficient ways of summing for finding the partition function. So if you did it naively, you would be stuck because it's an exponential, a sum with an exponentially large number of terms. But one can do it using matrix methods, and for some flavors of it, using recursion instead of matrices. And I don't want to give you the impression that we devised it from ground up. So like I said, Perkus in the physics department at NYU did it many years ago for liquids in some form. And then people took it over to Chromatin some years later, but everybody took a slightly different path. I would say this is the most comprehensive framework. It has the 2-body interactions, multiple species, but components of it certainly exist with other, in other labs, other publications. Okay, so The total energy, as I said already, can be given by any model you want. This particular model that I will describe a little bit has a sequence-independent offset, like the total binding energy, and then sequence-dependent corrections that are mononucleotides and dinucleotides. But again, I'm not, this model was easy to implement, and we showed in one paper that it works well as many other more complex models. But things change, and it depends, if you have a more complicated model that can be put in as well. So yeah, but the right thing to think about is that you have some energy to bind. Like I argued that this is maybe 18 kT, and then maybe plus minus due to sequence, right? Plus minus 3, let's say. This is this 18, and this is the sequence-dependent effects, which are kind of secondary in this picture. Okay, so forward solution, meaning from energies to occupancies, can be done on multiple species as well. So which was our goal all along. In a full model, you have to deal with matrices that are lengths of DNA by the number of species. So if the number of species is T and length of DNA is L in base pairs, it's LT by LT matrices. So if you do it on a whole chromosome, you're dealing with like 1 million by 1 million or 10 million by 10 million. That's maybe not practical, but for a piece of DNA is okay. So moreover, like I said, sometimes you have a recursive solution, but you have to unfortunately throw out the long range, the two-body effects. If you do that, then you can get rid of the metric thing and just do a recursive update, which is linear in the number of base pairs. You can still do multiple species, just no talking to nearest neighbors. So all of this in this PNAS paper. So then there's the inverse solution that basically tells me you give me a nucleosome occupancy profile and I want to infer energies. So and then you can fit any of the energy models you want, right? So this is an inverse, which is usually harder than forward. So this is interesting because you do an M&A's assay and then you get a big but noisy profile, and then you put that in and you get some energies that you can try to fit your model with dinucleotides on. Instead of trying to guess and do it forward and then do a least squares fit or something like this, do it many, many times, you just go backwards once. Okay, and when there are no long-range interactions, again, two-body is tricky. So you can do a recursive update. Doesn't really matter what's going on here, but basically you build partial partition functions. So, partition function of length I to the end is the previous one times some factors which are easily computed. So these are basically the probabilities, the experimental probabilities of finding a particle at an edge. So these are basic things. This is a thing you can easily compute from basic things, and then you can build the partition function recursively. And then once you have these objects, the z pluses and z minuses, and z plus or z minus for the whole length is z, the full partition function, then it turns out that you can compute the difference between the binding energy and the chemical potential with a log of the product of some of these things. So this is an exact statement. It's not an expansion or something like this. But of course, if you have noisy data, you estimate it from where the reads are, right? Or some model where you denoise the data, then you won't have, it won't answer, it won't give you a perfect energy, but it will give you perhaps an energy profile to compare with your favorite profile that's subject to experimental noise, right? So the idea that I wanted to sort of put into your heads is that all of this, the forward and the backward, it can be done. The simpler versions are linear in the number of DNA base pairs, therefore they can run on yeast all chromosomes, I don't know, in 1 minute. And you can go from your M&A sequence profile experiment plus some pre-processing to energies, which you can then interpret. Or if you stick to your best DNA bending model or something like this, you can run it forward and see if your occupancies look anything like the true occupancies that you see. And then if they don't, you can argue it's the experimental artifact or just your model is bad. I don't know. Yeah? No, it's fine. Yeah, go ahead. I'm almost done. sorry, how do you have two populations? Oh yeah, yeah, yeah. Oh, if you combine, yeah, if you combine, then you get some sort of average picture. If you do them separately, you can appreciate the differences in the free energy profile. Yeah, it's more. Yeah, the only reason not to the only reason to combine is to cut down on the noise. And if they both are good enough already, I wouldn't combine them because you will. That's true. And this, so well, if you leave it in for too long, then you will have, a profile that may not have anything to do with, say, sequence-dependent nucleosome positioning. The hope that people always have is that MNAs is done well, like they stop it before it eats into nucleosome. So we've also done, but I'm not showing the data because, well, I'm out of time. But the MNA's concentration can go from low to high, and you can do this, and you can try to work with multiple profiles like that. But it's full of artifacts. If you leave it for too short, you will have multiple nucleosomes, like pieces, with 2 and 3. If you leave it for too long, it will chew into the nucleosomes, for sure. You can see it on a gel. So let me show you actually not MNAs, because this is lower quality. But this experiment, again, due to John Wyndham, where you actually use hydroxyl radical footprinting to find out center-to-center distances, more or less, of the neighboring nucleosomes. So this is where this model shines, because this gives you not just the length of a single particle, but the distance between neighboring particles, which our model is able to sort of take into account explicitly. So these are the details, and yeah, let me just skip that. And what you get is basically the distance between neighboring diodes. Okay, so, well, there are experimental details that I'll gloss over. It doesn't cut diode to diode, it cuts nearby, and you have to kind of average over that. But basically, you have an ensemble like this, and if you just plot, which is their figure, right? If you just plot them, the distribution of these lengths, you will see that a lot of them are less than 147, which means that the nucleosomes are too close. So this also depends on how soon they stop the experiment. Nothing is perfect. It's a little bit like MNAs. But if you look at the below 147 on the blue one and on the red one, it's still significant. Right? So many of them are just too close. So that was a surprise to people. So because the standard, in Gary Felsenfeld's, you have bits on a string, right? Something like this. And this happens, but just not always. Sometimes it happens that they kind of crowd and invade each other's territories. And apparently, so I talked to Sergei Grigoriev yesterday. He left for today, and he sees this even in his in vitro experiments on short fibers. So, but this is on yeast. So using this two-point information. And our idea was then to fit the potential so that we can reproduce this using, of course, the model with unwrapping, because without unwrapping, you cannot proceed. And this is so we tested many potentials. So this is the unwrapping potential. Okay, so the sequence dependence is there, but it's secondary. This is just the unwrapping potential. And we tested many models, just a flat and so on. And this is the winning one, not a flat, but just a straight line, which is actually matched to the crystal structure. So the period is approximately 10, 11 bases. And then what's fitted is, I think, two parameters, the slope and then the height of the oscillations. Okay, so this was in the PNAS paper. And then we're able to reproduce. This X-ray means that it's informed by the crystal structure. It doesn't mean anything else. So this is the data. The data was just from before. I was showing you the experimental distances. It fits by the model well. And if you don't do unwrapping, you get some sort of null model which doesn't work at all, which is not surprising. Mm-hmm. Yeah, no, I just don't remember because we tested many models. For some of them, it does it only weekly. So we have a huge supplement in that paper, and probably some of it is there. But yeah, but we don't have any sensitivity. It's a good question. Yeah. That's true, but the phase here is fixed by the crystals. So I don't think we messed with the phase. We may have in another model. So certainly you can mess with oscillation, slope, and the phase. The phase should have a lot of effect, and I kind of vaguely remember it did. But this one, I think we just did the phase on the crystal structure because it says X-ray. But we had another one which is just a pure oscillation, and there I think we changed the phase, and I vaguely remember that it mattered a lot, as it should. For heights within some range, maybe it was okay. And the slope, I think, had to be pretty, this is unwrapping energy per base pair, so that had to be fit. But I guess that was a huge paper already, but we don't have the sensitivity. we could have, because these are all not expensive. Yeah. Mm-hmm. You could put any model in there, and it's efficient. Yeah. So okay, so that's it. This slide doesn't mean it. It just means that I've been trying to work with chromatin remodelers and kinetics, and this is hard. Thermodynamics, I would claim, is done. There is not much else you can do along these lines with one-dimensional systems, right? You can do 3D simulations. That's a different kind of soup, but for kinetics, translocation, especially in the presence of chromatin remodelers, you need some sort of Fokker-Planck treatment. Even at steady state, it's kind of tricky. And there are some papers that are appearing now, and one came out sent to me personally by Tom Chu from UCLA. And he did a long paper with kinetics on the mesoscopic scale. So it's not impossible, but there's also like lack of data as well. You need on-off rates, and if you fit all this stuff, it's a crazy fit. And it's, still, I think something is doable on these lines. We're trying with people from CNIS, Lyon, Cedric, and Benjamin Audit, and people like that for a while. Okay, that's it. So I just wanted to say that at the time it came out, it was a surprise that they're so crowded and even like invade each other's territories. So we have this thermodynamic model that was in MATLAB first, and now I recoded it in Python, which is more or less readily available if people want to collaborate. And then unwrapping is important. It was a surprise at the time first published. Now it's less of a surprise. Everybody talks about unwrapping. And what we're trying to do is ATP-dependent remodelers and basically kinetics of nucleosomal arrays at the moment. Okay, that's it. This is just a slightly out of date version of my lab with people went to all kinds of places. So the person mostly in charge of this project was Razvan, and Razvan is right here. Brilliant student. Unfortunately, he went to a hedge fund after trying to find a good position for a number of years. It's very good. His wife, who is a mathematician, is also there. So not good for nucleosomes. Well, thank you very much.